Hello, everyone. Okay, thank you for coming back. I hope you all enjoyed your lunch. I have to tell you that this is your last chance to vote for the best lightning talk and the best poster. The moment Dr. Beasley starts talking, voting will close because we need to have a little bit of time to calculate the scores and everything. So make sure that you have used Slido to vote for the best poster and the best lightning talk. Thank you for that. So the afternoon session has two presentations from two of our faculty members on cutting-edge research in understanding the pathophysiology of mental disorders, but also improving the treatment of mental disorders. So the first presentation is from Dr. Claire Beasley, who is also part of the planning committee for this meeting. She's an associate professor here at the Department of UPC, and she heads her own lab, focusing mostly on post-mortem research in patients with bipolar disorder and schizophrenia. The aim is to identify molecular pathways that might be involved either in the pathophysiology of the disorder or in medication use. Today, she will specifically focus on the complement. It's a component of the inflammatory response that has been implicated in psychosis, and she will give us an overview of the topic and of her work. Dr. Beasley.
Can I? Okay, thank you. Thank you, Sophia, for the introduction. As you just heard, one of the main areas of interest of my lab is the role of the immune system in psychiatric disorders, which we investigate primarily using human postmortem brain tissue, but also peripheral samples, including blood and saliva. And today I'll be talking about some of our ongoing studies looking at the complement system and the etiology and pathophysiology of schizophrenia. I don't have anything to disclose. So in general, there's been a lot of recent interest in the role of the immune system in schizophrenia. But this really isn't a new idea. So going back to even 1911, Bloiler suggested that the connection of schizophrenia to the infectious process needs further study. And at a similar time, Kraepelin suggested that infections in the years of development might have a causal significance. So even going back over 100 years, there was some interest in the role of the immune system in schizophrenia. And over the past couple of decades, really, there have been studies, epidemiological studies, biochemical studies, and genetic studies that have really borne this out and suggest that the link may be there. So schizophrenia is associated with an increased rates of viral, bacterial, and parasitic infections. Schizophrenia is also associated with higher prevalence of some autoimmune disorders, but not others. And conversely, individuals with autoimmune disorders do have a higher risk of developing schizophrenia. In addition, there are increased risk of schizophrenia in individuals whose mothers had a maternal infection or maternal autoimmune disorder during pregnancy. And then moving on to sort of studies of blood, it's been shown that there are alterations in immune cell parameters, including increased neutrophil to lymphocyte ratios in individuals with schizophrenia. As well as that, there's increased levels of some inflammatory mediators, in particular some cytokines. So meta-analysis have shown that there are increases in C-reactive protein, CRP, as well as cytokine IL-6 in individuals with schizophrenia, although this evidence is quite mixed. But some of the more recent evidence linking the immune system with schizophrenia comes from genome-wide association studies. So here you can see this is a Manhattan plot, and this is from the most recent genome-wide association study in schizophrenia, which was published last year. And you can see that this involved 75,000 cases of schizophrenia, 240,000 controls, and they found associations at 287 distinct loci. So just trying to orient you to this plot, on the x-axis is chromosome, and on the y-axis you can see the p-value. And each of these green diamonds represents a distinct loci that's been associated with schizophrenia risk. So the main ones I've labeled here, the first one, the very large peak you can see on chromosome 6, is located within the major histocompatibility complex. And this is a region of the chromosome that contains a number of genes that are involved in immune function. And in particular, fine mapping of this region has suggested that a significant portion of this signal is really produced by variations in the complement C4 gene. As well as that, another complement-related gene, CSMD1, was also found to be a risk factor for schizophrenia. So this has really sort of piqued folks' interest in the complement system in schizophrenia. And really since this time, there's been a number of studies that have really been looking at the complement system. So I just wanted to really focus a little bit on what the complement system is. So this is quite a complicated diagram. I'll walk you through it. The main role of the complement system is really to complement the rest of the innate immune system. But it's involved in phagocytosis, so particularly phagocytosis of so pathogens or cells that are undergoing apoptosis or cellular debris, things that shouldn't really be around in the brain. It also induces an inflammatory response. So within the complement system, there are three main pathways. The first one is the classical pathway. So I'll just sort of go through this. I don't know if you can see the pointer. So the classical pathway is activated when C1q binds to a pathogen or an apoptotic cell, or in the CNS, even a synapse. And this leads to the cleavage of C4 and C2. So C4 is cleaved into C4a and C4b, with the C4b joining C2a, and that forms the C3 convertase. And then this kind of has a sequential action. So this cleaves C3 protein into C3a and C3b, and the C3b part joins on with a C3 convertase to form C5 convertase. And then subsequently, the C5 convertase cleaves C5 into C5a and C5b, and the C5b part joins on with a bunch of other complement proteins, C6, C7, C8, and C9, to form the membrane attack complex. And this is the sort of final effector pathway. It leads to lysis of cells, causing the cell death. There are two other parts to the complement cascade. So there's a lectin pathway. This is essentially analogous to the classical pathway, but it is activated under different circumstances. So it's activated when MBL binds to specific carbohydrates on cell membranes. And then there's also the alternative pathway, and that's sort of normally active at a very low level in the essentially it's activated due to spontaneous hydrolysis of C3. Okay, so just to point out some of the most important components of the complement cascade. So you see here highlighted in red, these are the opsonins. So these are essentially the members of the cascade that bind to pathogens or synapses or apoptotic cells, and they essentially tag them for removal. So these cells that are tagged or synapses are recognized by receptors in the brain. These are particularly recognized by receptors on microglia, and then microglia come and phagocytose the debris or whatever the target is. In addition, there is C4a and C3a, which are anaphylotoxins. These actually sort of produced further inflammatory responses. So they kind of activate other parts of the immune system. And in particular, they produce molecules that sort of cause the phagocytic cells to come and find the targets. And then finally, there is the membrane attack complex, so C5b9. And as I mentioned, the role of that is to cause a pore in the membranes of cells or sort of other things, so potentially synapses. There's some recent evidence that suggests that the membrane attack complex also can form pores in synapses. And this obviously is the final pathway of complement activation. So we know that most complement protein is produced in the liver, and it doesn't actually get through the blood-brain barrier into the CNS under normal circumstances. However, recent studies have shown that cells within the CNS are capable of producing complement as well as complement regulator proteins. So this is sort of quite interesting. I mean, there's been a lot of questions. Why would the brain have this capacity to produce large amounts of complement proteins when there's very little sort of infection or anything like that going on in the CNS? So recent studies have indicated that, as well as playing a role in the immune system, a lot of complement proteins also have different roles in CNS, particularly during CNS development. So complement proteins are particularly involved in neurogenesis, neuronal migration, but also synaptic pruning, synaptic plasticity. So here you can see on the right-hand side, this is just a cartoon, the large green cells in microglia. And you can see here that this synapse here is being tagged by C1q and C3b. It could also be tagged by C4b. And then the receptor on the microglia, the CR3, comes and recognizes the complement protein and phagocytoses the synapse. Well, most of the evidence so far suggests that this process is ongoing during brain development. There's also some interest in the role of this sort of complex mediated synaptic removal during aging as well. And there have been several studies in neurodegenerative disorders that suggest that synaptic tuning in disorders such as Alzheimer's disease is mediated through complement. So in terms of schizophrenia, obviously, as I mentioned, some of the most compelling evidence comes from genome-wide association studies. Obviously, this large peak in the latest genome-wide association study is predominantly due to the complement C4 gene. And this has really, as I mentioned, led to a lot of interest in the complement system. And there have been quite a number of other studies that have provided evidence that complement may be altered in individuals with schizophrenia. So, for example, there have been studies that have quantified C3, C4 protein levels, as well as the activation of the complement system in blood. Some of these studies have found increases in C3 and C4 protein levels, although studies do vary somewhat. Not all studies have found this activation levels to be increased. Other studies have looked at mRNA expression of C4 in particular, and there is evidence for increased C4 mRNA expression in brain, in CSF, and in blood as well. And also, there have been more recent studies looking at CSMD1 since the latest GWAS study. And it's been shown that CSMD1 mRNA expression is actually decreased, in particular in first episode psychosis. As well as that, there have been some studies in animal models, and studies in animals have shown that if you do increase C4 levels, that you see lower levels of synapses and dendritic spines in animals, as well as behavioral changes, in particular impaired working memory. And it's been suggested that animals with increased C4 expression may be a potential animal model of schizophrenia. Okay, so moving on to our studies in the lab, obviously there's a great deal of interest in the complement system right now, but not a lot is known about the effects on the brain of complement, or whether or not levels of protein or mRNA expression are changed in the brains of individuals with schizophrenia. So that's essentially what my lab has been looking at over the last couple of years. So essentially, we've quantified complement-related proteins and mRNA expression. in postmortem human brain tissue. These samples from the Stanley Foundation in the US, and they comprise 35 individuals with schizophrenia, 35 controls. So we also looked at 34 individuals with bipolar disorder, just because this was part of the same brain series as well. Protein expression was quantified by Western blotting and gene expression by real-time PCR. And these are the same subjects that we quantified protein and gene expression. So this is just an overview of our sample. As you can see, the age is quite similar between groups, so around mid-40s. And we have quite a lot of information. So we have information on things like brain pH, BMI, sex, cause of death. So you can see the list there. The thing that we don't really have is information on symptoms. So we don't actually know whether or not these individuals were experiencing positive or negative symptoms, cognitive impairment, unfortunately. So I'm just going to go through some of our data. Obviously, these are ongoing studies, so I only have data for some measures, not all measures, but we are sort of working to sort of go through the whole list of complement proteins and their regulators. So I want to start with C1q and C3. As I mentioned before, these are the opsonins. So these are the sort of molecules that bind to either synapses or cells to regulate the phagocytosis. And we didn't actually find any difference between groups for C1q mRNA levels, C3 mRNA levels, or C3 protein levels. So you can see here we have the controls on the left in the yellow bars, the schizophrenia group in the middle in the blue bars, and the bipolar disorder group on the right-hand side in the purple. So then we moved on to look at C4. Obviously, this is the gene that most people are most interested in. C4 is actually quite a complex gene. So it comes in two forms, C4a and C4b. And then within that, there are long and short versions of the gene, depending on the presence or absence of a human endogenous retrovirus in intron 9. As well as that, individuals can have different copy numbers of C4a and C4b. And there was a study back in 2016 that looked at C4 haplotypes to see whether there was any difference in the risk of schizophrenia. And you can see here that individuals with the long form of the C4a gene were at increased risk of schizophrenia, whereas people that had the B form, the B short form, weren't actually at increased risk. So there's obviously a difference between the A and B sort of forms of the C4 gene. And similarly, when they looked at C4 expression, obviously individuals with higher copy numbers of the A form, in particular the A long form, had higher C4 expression. And in terms of the protein, the C4a and C4b proteins are actually almost identical. They only differ at 4 amino acid residues. So it's kind of hard to tell them apart, but despite that, they do actually have quite different functions. So the protein that is derived from C4a is mainly involved in immune clearance and opsonization, whereas the protein that's derived from C4b is more involved in activation of the complement pathway as well as cell lysis. So sort of in the longer term, it is quite important that we are looking at the C4a and C4b proteins separately. So far, we haven't done that due to a lack of antibodies, but that is something that we do plan on doing. So in terms of C4b, as you can see here, we found a significant difference between groups for C4a mRNA expression. We found higher C4a mRNA expression in individuals with schizophrenia. And this is consistent with the prior study by the Makawa group. In terms of protein, while we did find correlations between C4 mRNA and C4 protein levels, we didn't actually find a significant difference in total full-length C4 protein between groups. Looking at C4 copy number, we were able to obtain C4 copy number for each of our samples. And as you can see here, really as expected, with increasing copy number, you do actually see an increase in C4a expression. But also with increasing C4 copy number, you also see an increase in C4 protein levels as well. So that suggests that obviously in individuals with schizophrenia, if they have increased C4a copy number, then that would also lead to increases in protein levels, even in the adult. Okay, so this is quite complex, but I just really wanted to point out that obviously it's really important to be able to regulate the levels of protein in the complement system. You don't want a sort of massive increase in C4b or C3b in the brain, because obviously that's going to tag sort of cells or synapses for degradation. So there are a whole variety of proteins that are involved in the regulation of the complement pathway. So as you can see on the left-hand side, we have the C4 protein. On the right-hand side, we have the C3 protein. The protein C1 inhibitor, or SERPING1, is really important in that it inhibits the degradation of C4 into C4a and C4b. And as well as that, there's a number of proteins that are involved in the degradation of C4b and C3b. So these include factor I, factor H, C4 binding protein, CD35, as well as CSMD1, which obviously is the other protein that was found in the recent genome-wide association study. And as I mentioned, these proteins are involved in the degradation of the opsonins into sort of further sort of breakdown products that aren't sort of able to cause phagocytosis. In addition, I've listed CES6 here. You heard about that earlier from Sia. So it's quite interesting. There have been a number of proteins that have been found recently to be involved in complement regulation that are actually really highly expressed in the brain, and also thought to be involved in synaptic plasticity. So we know that some of these, there are knockout models, and there are sort of deficits in synaptic and dendritic spines in these animals. So we are sort of quite interested in finding out a bit more about what's going on with these proteins as well. So we did quantify mRNA expression and some species protein levels of these complement regulatory molecules. So first, looking at C1 inhibitor, we didn't find any change in C1 inhibitor mRNA, but we did find a significant increase in C1 inhibitor protein levels in the brain in individuals with schizophrenia, as well as in bipolar disorder. And this suggests that this may be potentially a compensatory mechanism in order to prevent the formation of the opsonin C4b. We also looked at factor H and CES6 mRNA. As I mentioned, these are the proteins that break down the C4b and the C3b molecules. We found no change in factor H, but we did find a significant decrease in CES6 mRNA in individuals with schizophrenia, as you can see here. We also looked at CSMD1 and CSMD2. We didn't find a significant change in CSMD1 mRNA expression in individuals with schizophrenia, although levels were lower, and it wasn't statistically significant. But we did find a significant difference in CSMD2 mRNA levels in individuals with schizophrenia. We don't really know exactly how CSMD2 impacts the complement cascade at this time. However, it has been implicated with schizophrenia before. And in fact, the major study run by Bob Stowe found a missense mutation in CSMD2 in one of their patients. So there is certainly some interest in looking to see what CSMD2 may be doing in the brain and how this is related to schizophrenia. And then finally, we looked at C9 and C5b9, which are obviously the sort of the final pathway of the complement system involved in the production of lytic pore. And we found significant increases in both C9 and C5b9 protein levels in individuals with schizophrenia. It's a little bit tricky to interpret this, but potentially this could indicate that there is activation of the complement pathway in individuals with schizophrenia. So obviously there are some sort of potential confounders that we should consider when we do postmortem studies or any kind of studies really. But so the first one is effects of sex. So there have been quite a few studies recently that show that the complement system is more activated in males compared to females, and that there is higher levels of complement C3, complement C4, C1q in males. I'm not quite sure why this is, but our studies actually, interestingly, did show that there was a difference between sex. So in our original models, we didn't find a significant effect of sex, but considering these previous studies, we did look at male and female cohorts separately. And interestingly, as you can see here, we found that C4 mRNA expression was only increased in males in schizophrenia compared to male controls. Similarly, C9 protein levels are only increased in male schizophrenia subjects compared to male controls and not in females. And this actually was across the board. So anything we found significant, so C5b9, CES6, all of them were only increased in males and not in females. We looked at some other confounders. Obviously, age is a big one. There is evidence that the complement system is increased, or the activation of the complement system is increased with age. This is something that we are quite interested in the lab. We're kind of interested in sort of accelerated aging in psychiatric disorders, and potentially complement could play a role in that. However, we didn't actually find any correlations with age in our sample between any of the complement proteins or mRNAs. Similarly, antipsychotic dose. So there are some studies that have shown that antipsychotics alter complement activation. We weren't able to find that in our subjects. But as you can see on this slide, there are some confounders that did seem to correlate with complement levels. So in particular, we were able to access serum CRP levels, and some complement measures correlated significantly with serum CRP. As well with BMI, we found some correlations between complement proteins and BMI. And this potentially suggests that peripheral inflammation may be impacting brain complement levels. And as well as that, we found some relationships between complement levels and suicide. And it's not quite sure what the cause of this may be. There has been some previous evidence that suicide is associated with inflammation. But this, I think, deserves further exploration. And then in addition, we looked at the correlations with synaptic proteins. So on the left, you can see a cartoon sort of showing the presynaptic site and the snare complex. So we were interested in this protein SNAP25, which is present in all sort of presynaptic terminals. And we quantified this in the same subjects using ELISA. And we were able to find significant correlations between SNAP25, so the presynaptic marker, and several complement measures. So in particular, we found that there were correlations with C3, C4 expression, as well as some of the regulators. And these were in a negative direction. So this really suggests that if you have more C4 protein or C3 protein, you have less synaptic terminals. So just to conclude, so we did find that C4a gene expression is higher in schizophrenia, in particular in males, and that while C4 protein levels correlate with C4a gene expression, C4 protein wasn't changed in individuals with schizophrenia. We found that C4a gene expression and C4 protein levels were associated with copy number. And there were also some changes in some of the complement regulator proteins in schizophrenia. In addition, we found increases in C9 and C5b9 in schizophrenia, which obviously suggests that there may be some cell lysis going on, and this could be potentially related to an increase in apoptosis. And then obviously, as I just mentioned, there are relationships between complement proteins and levels of SNAP25, which is a presynaptic marker. And overall, I think our data is consistent with the role for complement in schizophrenia and potentially through the tagging of synapses for removal. And finally, we do have obviously some future questions. We are kind of in the middle of the study still. So the first one is that we only investigated total C4 protein levels, and obviously it would be interesting to look at this protein that's derived from the C4a isoform, as well as looking at C4b and C3b fragments, which are the sort of functionally important fragments of the C4 and C3 molecules. And this is something that we are doing in the lab right now. Another question is whether complement opsonins are actually deposited at pre or postsynaptic sites in individuals with schizophrenia, and whether this is potentially sort of preferentially at excitatory or inhibitory synapses. And that, again, is something that we are interested in looking at right now. There's also a question about when complement disturbances may be manifested. And it's obviously quite difficult to look at this in human postmortem brain tissue. Our samples are, as you saw, in their 40s predominantly. And it is kind of hard to look at earlier ages. And one thing that we are sort of working on right now is to try to get some methods up and running to look at extracellular vesicles. So one idea would be to try to see whether we could look at first episode patients. So extracellular vesicles are essentially vesicles that are released from brain cells. They cross the blood-brain barrier, they get into the serum, and they contain proteins that were derived from the original cell that they came from. So we can actually look in blood at proteins that were coming from brain cells and quantify complement levels in blood using that method. There's also questions whether complement dysregulation may be associated with particular symptoms of schizophrenia, and there's some evidence that it may be associated with cognitive function, in particular working memory. And obviously, there are some questions regarding specificity. So alterations in the complement system aren't specific to schizophrenia. They're present in a whole bunch of different psychiatric disorders, as well as in sort of immune disorders as well. So I think it's really important to try to figure out specifically what may be going on in schizophrenia, you know, in light of the GWAS findings for C4. And that could potentially lead us to some ideas about not only the pathophysiology of the disorder, but also future treatments. And okay, to conclude, I just want to thank the folks in the lab that actually did most of the research. This study is funded by CIHR. And I would like to give a plug, finally, to anybody that is doing research in schizophrenia that would like to join us. We have a meeting of the Canadian Network for Research in Schizophrenia and Psychosis, be held in Vancouver on October 17th and 18th. And if anybody's interested, please let me know and I can provide further details.
That was wonderful, Claire. Fantastic, very logical kind of and complete hypothesis testing. Really beautiful. Questions? Is one person there? Yes. That's you. Yes, you're the person.
Great panoramic talk. I just have a question for your intro material. When you talk about complement being important in development versus actually normal development, we know when, like is it prenatal infancy, like where would you place that somewhere?
Yeah, I think it's important across the board. It's definitely important prenatally in terms of migration of neurons. But then I think we sort of don't really have a full answer to that, but it is certainly important across the whole lifespan, really, I think.
And sort of a follow-up, is there any thought you're looking at how post-mortem brains that there could have been a perturbation in the complement system, a big signal earlier that we just can't see? You're looking at way downstream, and how would we ever get to that?
Yes, absolutely. I think that's a really important question. Obviously, there's so many things that have happened to these individuals by the time they've sort of reached us. So certainly we would be very interested in looking in sort of first episode patients. Obviously, that's sort of more proximal to the time of sort of synaptic pruning and that kind of thing in frontal cortex. But it is quite difficult to look earlier than that. I mean, it's kind of a little bit tricky. I mean, there could potentially be animal models that would help, and we can look through the lifespan there. But certainly, I agree with what you're saying, and that's something that we would be sort of very mindful of trying to find ways of figuring that out.
Thank you for the great talk. I was wondering if you or anyone else has looked at whether the complement activation and synaptic pruning that happens is more specific to specific types of synapses, for example, excitatory, inhibitory, specific neuronal types, or anything?
Yeah, that's a good question. There's some evidence that it may be more in excitatory types. There's a lot of studies recently in animal models that suggest that the activation sort of in inhibitory types acts through astrocytes rather than microglia. So that's something that we would really be interested in doing in schizophrenia, to see whether this tagging does occur predominantly in excitatory or inhibitory types of neurons, but I don't think we know that right now.
Sorry, a follow-up to that. Do you think it would be possible to do the same method that you did with the SNAP25 with, for example, specific excitatory markers like V-GLUT or something?
Yes, absolutely, and that's what we are planning on doing in the next few months.
I don't have access to Slido. Team, can you see if there's any question from there? Sorry.
I don't actually have it up right now.
Oh. Perhaps, is there a question? So while we're doing it, I will ask a question. So is it possible to do this research from CSF, or at least part of it, complement levels and so on?
Yes. Yes, there have been some studies in CSF. Yeah.
Because in Germany, until recently, all first episode patients had a CSF taken and examined for, and the benefit of having the CSF is you can look at other markers of immune activation if they are present, but potentially you could link up with some of the German colleagues that still do that.
Yes, that'd be really interesting.
They do spinal taps. No, nothing from Slido. Anything else from here? One last question. Yes, the one at the back.
Hi. So I thought it was really interesting how you talked about accelerated aging as a possibility, and how the protein levels or mRNA levels didn't associate with chronological age. And I think that you would need the data for this, but it made me think of whether these levels would associate with a biological measure of age. For example, epigenetic age acceleration, or even like telomere lengths and such. I think that would be really interesting.
Yeah, that's a good question. I think that would be really interesting too. I mean, we do have some studies in the lab looking at biological markers potentially of accelerated aging. Unfortunately, CHR didn't give us any funding to look at sort of telomere length, but certainly it would be something worth looking at. Thanks.
Thank you.
Thank you very much. Okay, so our next talk is from Dr. Joseph Pham. He's probably well known to all of you. He's been a member of our department since 2003, and he's involved in multiple different and very important leadership roles. He's currently the ECT coordinator at the UBC Hospital, the physician lead in the older unit at St. Vincent's Hospital, the associate medical manager for the BNC Neuropsychiatry program, and he's the clinical faculty representative within the executive committee of the department. And his general sort of interests are very wide, but mostly focused on neuropsychiatry. and more recently on the effect of different psychedelic or other kind of types of rapid acting antidepressant molecules, and he's spearheading really the development of this type of treatment within UBC. So we're very much looking forward to your talk.
Thank you very much, Sophia. It's a pleasure being here. First time I've actually got like a, in a real life talk since the pandemic. Most of the time you're looking at little square boxes. So it's a pleasure and a blessing to be here today. Let's talk about ketamine and psychedelics today. Let's do a little bit of clinical research to close off today. First off, no disclosures of real significance. I don't have any financial interest in this, but I will be talking obviously about an off-label use of a medication. It's an anesthetic medication, but for psychiatric purposes, not officially supported by the FDA or Health Canada. Now, the objectives are, just quite straightforward. First of all, with about 30 minutes, let's just quickly talk about the psychedelic drugs, what they are, just broadly, and then just very broadly, how they work. Secondly, let's jump into ketamine itself. Let's talk a little bit about the background of this drug, how it came to be of interest in psychiatry, and then a little bit about how we're using it. And then finally, a bit more about the clinical role that it might play and how we can think about this in the days ahead. Now, unless one lived under a rock, you could not avoid all the talk about psychedelic medications in the last few years. It's sort of like you can't escape from ChatGPT talk as well. So whether it's in the news media, whether it's CBC or Globe and Mail or in the videos or on YouTube, you've probably seen something about psychedelics. And in fact, you can even look on websites for major hospitals. For example, Johns Hopkins has a psilocybin research arm as well. Now, you can imagine that psychedelics are capturing the imagination because that's literally what they do, right? They are psychedelic means they are mind manifesting. They have the ability to distort our spatial or temporal experience. There's a subjective, at least, expansion of our sense of consciousness. And then, of course, within the spiritual religious realm, you have all kinds of terms about this. There are the opportunities for a transcendent spiritual experience, for meaning, awe, holiness in our experience of life. We've been here before in terms of a research, in terms of research endeavors. So, for example, if we look at this slide, this is a look at PubMed publications of all research involving the term psychedelics over the years. It looks something like this. Back in the 1940s to 1960s, let me see my slide. Let me see if I can turn on my pointer here. Okay. See that? Okay. So from basically about the 50s all the way until about 1970, psychedelic research was something that a lot of people engaged in all the major universities in Europe as well as North America. Back in those days, the Sandoz company produced Delicid, which is LSD, as well as indocybin, which is psilocybin, available for research purposes. By 1970, however, the psychedelics became a Schedule I Schedule I substance, basically said to have no significant medical purpose. And from that point on, we entered this quiet era of psychedelic research through the 70s, 80s, 90s, until about 2000 or so. In the year 2000, that was the first publication of a placebo-controlled trial using ketamine in the treatment of depression. But even until that, but leading into that time, there were also other interests in terms of how the drugs could be useful in psychiatric conditions. Now, that last slide with PubMed basically look at all psychedelics, right? And this is a quick chart of the four major classes of psychedelics that we have to think about and consider when we look at, the drugs and use in mental health. There are the old anticholinergic type drugs like atropine, atropine belladonna, for example. These drugs, being severely anticholinergic, can cause delirium, and in that delirious state can cause auditory hallucinations, visual hallucinations, as well as other symptoms. Then there are the catecholamine drugs, the dopamine, noradrenaline. And and there's a lot of cross-reactivity, so there's 5-HT involvement in these drugs as well. MDMA being kind of the big one in this group, that's currently going through phase 3 trials, looking for FDA approval for treatment of traumatic experiences, for example, being a very common indication. Then there are the indolamines. These are the classic psychedelic drugs, LSD and psilocybin being the two big ones, although of course, ayahuasca is often talked about within the spiritual literature based on the South American herbal type remedies. And for us today, we're going to focus on the NMDA antagonist drug. These are PCP, phencyclidine, angel dust, and ketamine, which of course is used quite frequently in our anesthesia world. So how do these psychedelic drugs work? Now, obviously, each of those classes will have their own receptors that are affected by the drugs, right? So there are different neurotransmitters, different receptors, as well as different locations. And these last two, very important, because whereas I'm talking about ketamine today and the psychedelic experience that some individuals might have with ketamine, there is, of course, another drug available to us that's also an NMDA antagonist. And that's memantine, which we use in severe dementia illnesses, right? The effects are very different, even though they are both NMDA antagonists. The answer, or at least we believe the answer, is probably got to do with the population of receptors and the locations. Whereas ketamine directly acts at the synaptic level, memantine actually is extrasynaptic in most of its location of action. Now, there's probably functional connectivity changes as a big part of this. I mean, at the big level, beyond the neurotransmitters and the receptor level. So, for example, research in the default network suggests that there's reduction in connectivity of the default mode network. And in that state of the psychedelic experience, there's actually increased connectivity of other areas of the brain. There's been talk about increased network entropy through the brain during the psychedelic experience. There's a potential for disruption of those stereotypes, those networks that we're kind of stuck in, perhaps in the depressed individual who is stuck in that negative cognitive network. And there's potential for structural change, synaptogenesis, even neuronogenesis, although for the psychedelic treatments, in psychiatry, we're using it for short periods of time. So it might not be so much that as the functional plasticity, as the ability to break some of those long-held network tendencies that the brain has. If you're interested in this kind of stuff, I would certainly recommend the book the journal article at the bottom there from Carhart Harris. This is from the Imperial College London. Great paper looking at evolution, neuroimaging, cognition, and even some Freudian analysis if you're interested in that too. All right, let's talk about ketamine, which is our low-hanging fruit when it comes to psychedelic treatments, and the one that has, I think, currently at least the most benefit for our patients in the early 2020s. Okay, what is ketamine? Ketamine was synthesized back in 1956. And just as an interesting aside, LSD was synthesized in 1938, same year as ECT. But look at how far behind in terms of the research that we have around psychedelic treatments. It's derived from PCP, ketamine, and PCP came out in the early 50s. But right from the start, we knew that PCP was not a very good drug for anesthesia because patients would have very severe, profound psychotic reactions as well as an anesthetic reaction. So they could be extremely aggressive and not feel the pain. And ketamine, therefore, became approved later on in 1970, the same year that the Scheduled Drug Act came into effect. So it escaped the rest of the psychedelic drugs in terms of being put into a Schedule I section of the Act. Ketamine is a non-competitive NMDA antagonist. And in sub-anesthetic doses, so if you go 2 milligrams per kilogram, 3 milligrams per kilogram, the person starts falling asleep. But if you give it at a lower dose, at around 0.5 up to 1 milligram per kilogram, there is this paradoxical reaction. The person actually becomes more disinhibited. Their energy level increases. They speak more, and they can also experience things in a different light. I just borrowed this quote from Crystal and Neuron 2019. So paradoxically, it increases the neuroplasticity despite blocking NMDA. There are various other downstream activities that it does. It will increase BDNF. It will change the mTOR system. But the key bottom line is that it disinhibits glutamate release cortically. So if we were to do, a functional scan during the experience, you would see increased cortical glutamate activity. In the 1990s, it was already known that NMDA receptor antagonism could provide some antidepressant type effect. So this was done in a mouse model using learned helplessness with various NMDA receptor antagonists. But it was this paper in 2000 by Berman and Crystal in biological psychiatry, where they did the first blinded trial using ketamine versus placebo. And it was a very small paper. It was only 7 cases. And these were patients who were 23 to 56, 2 weeks off all antidepressant type medications, and they were given 2 infusions, 1 week apart. One could be ketamine, one could be placebo, and they basically measured the mood reaction of the individual over the next 72 hours. And this is basically what they found: a very significant reduction in the mood score. In this case, they used the Hamilton Depression Rating Scale over 72 hours. How valid is that? How quickly can you actually judge, the time frame? Hard to say, but clearly, I think in clinical experience, when you see these patients, it's quite remarkable just how quickly the depression improves. And basically, the other scales that they used, for example, they had this Likert scale looking at whether the patient felt high or not. Clearly, it's very hard to blind ketamine treatment. The moment you give the ketamine, within an hour, you see the spike versus placebo, where the person doesn't really feel anything whatsoever. Regardless, that started the cascade of papers in ketamine. And as time went on, the opening of the discussion around psychedelics. Definitely by 2006, psilocybin research started becoming more and more in the literature as well. Okay, now it's difficult to, I just want to show you this other study. This study was from the American Journal of Psychiatry, 2016. And for me as a clinician, this is one of the most useful papers, because this is one of those that actually looked at serial treatment with ketamine 2 times a week or 3 times a week, and see what happened to the depression scale over the course of weeks. So whether the person was given twice per week or 3 times per week, basically they showed a reduction in the mood scale. And this is compared to the placebo group that they tried. Most ketamine, most of the research is around the dose of 0.5 milligrams per kilogram. Like I said earlier, if you push it too high, patients fall asleep. All right, so they continue antidepressants through this trial, which is what naturalistically we would do in the clinical world as well. And basically what they said was that there's no difference whether you gave it twice a week or 3 times a week. The depression reduction happens about the same. That within about 11 to 12 days, you basically hit the lowest plateau for the depression scale. And follow up after day 29, so a month later, they showed that the Montgomery-Åsberg depression scale remained minus 21 points below what it was before when the person started, which is quite remarkable. While it's difficult to generalize this data, the methodology is extremely different across the board, depending on which study was used. The response is somewhere around, and response defined by a 50% reduction in these depression scales, is somewhere around 50 to 70% in these studies. And the remission, that is the absolute value, like the MADRS dropping below 10 of treatment refractory depression, these are not naive patients who have not tried an antidepressant. Even treatment refractory depression patients, somewhere around 20 to 40% achieve remission of the symptoms. And during the pandemic then, while we were all at home thinking about what we wanted to do, myself and a few co-conspirators decided to put together a protocol for UBC Hospital. And I'll explain why we're doing what we're doing. This is a protocol for an inpatient treatment using subcutaneous ketamine. Which subcutaneous is used, but and all kinds of modalities are used, right? You can have intranasal, you can have intramuscular, sublingual, oral preparations, all within possibility. But for some reason, I just thought it's kind of unusual that subcutaneous is not used as much in the research around ketamine. Because subcutaneous is what we do, for example, with insulin since 1912. And there's a known process in which the medications get absorbed. So why consider subcutaneous or intramuscular as our mode of treatment? Well, basically because if you look at bioavailability of parenteral ketamine, it's very similar to IV, which is what people have been using for a long time. About 90 to 95% of the ketamine, when given subcutaneously and intramuscularly, eventually will end up in the system. And when you look at some of the studies, there are not many, and this is one area that I would love to have funding to actually explore, is that the release of the ketamine over the course of time with subcutaneous is actually quite similar to the plasma level that you would see with IV treatments. And it's relatively inexpensive. A bottle of ketamine for treatment in anesthesia is about $50. And we can do multiple cases with straightforward racemic ketamine that we can get through anesthesia when pharmacy allowed us. And it's also really pragmatic, right? You know, on a psychiatry ward, you're not going to be running IV medications. Most nurses are not trained in it. Infusion pumps are hard to get. So for all those kinds of reasons, it's much easier to do this in a subcutaneous way. So the key, though, of course, is to hopefully be able to demonstrate that the result is similar to what is out there with intravenous treatments. So this is basically, you know, just quickly running through the protocol. It goes through the inclusion-exclusion is quite similar to what most people do in the ketamine world. Basically, we want to treat patients who are treatment refractory, at least 2 antidepressants, plus 1 augmenting agent, quetiapine, lamotrigine, lithium, whatever it might be. We think of ketamine as an augmenter. We only use it for a short period of time. We have a permission to use it for 6 treatments in our inpatient protocol. So within those 6 treatments, twice a week, so 3 weeks, let's see if we can get patients' depression levels down and better. And if the clinician that sends these patients to us for inpatient treatment want to change the medication, let's do it. Because in the long term, 3 months, 6 months down the road, we're going to be dependent on the patient's daily medications. We got to watch for psychosis and substance use, of course. Ketamine is a substance of abuse in the community. Not very common, you know, maybe something like less than 0.5% of substance users might use ketamine, but it's certainly out there. And finally, as with, I think, all treatment refractory depression treatment type cases, we got to be mindful of cluster B, axis 2 type disorders, personality disorders. Doesn't mean that we can't treat patients with personality disorders, just that it cannot be the primary condition that we're dealing with that might be causing the low mood. All right, the treatment protocol was pretty straightforward. We wanted to make sure that it was easily replicable, easily doable, and we start at a low dose, 0.25 milligrams per kilogram. Part of it was my curiosity. Can even that lower dose than 0.5 milligram per kilogram, which is a standard starting dose, can even that lower dose help patients and expose them to less of the drug? And then from there, we do 2 treatments, and then we increase to 0.5 milligrams per kilogram for 2 treatments. And then if necessary, based on our measurement of the MADRS and the QIDS SR16 for depression, we can increase it to 0.75 milligrams per kilogram. Of course, all of that, you know, we had some nice discussions with our anesthesia colleagues, made sure everybody was happy, and, you know, we didn't likely weren't going to run into issues. So I'm going to give you the data for the first 24 patients that I've done on 1 West and 1 East here at UBC Hospital, just across the way. So since June 2021, when we started the project, I've done about 30 now, but this is the first 24 that I was able to collate the data. Here are the demographics. Basically, these were individuals, most of which were between 30 to 50 years of age. 13 men, 11 women. 22 of them had major depressive disorder. 2 of them had bipolar depression. On average, these individuals had tried more than 3 antidepressants in that current episode of depression. And on average, at least 2 augmenting agents were on their list of medications tried. On average, these individuals had 24 months of continuous depressive symptoms. So these are relatively severe patients that we had. Of interest, 11 of these 24 had tried ECT at some point in the past. And in fact, 2 of these patients were sent to me for ketamine treatment after just having ECT at least 12 treatments less than 2 months ago and still did not recover from the depression. 5 of them also had neurological conditions because I work on neuropsychiatry, and a lot of these cases tend to have previous strokes, multiple sclerosis, one with Parkinson's disease. And this is the graph that we have, and basically this is like what you saw in the previous graphs with the IV ketamine. So on average, there was a 48% reduction in clinician administered Montgomery-Åsberg score within 24 hours of the treatment. And remember, we start at a low dose, 0.25 milligrams per kilogram. I've got to admit that I get quite, I was quite surprised because sometimes patients will tell me, oh, I don't even feel anything. But yet after a weekend pass, they come back and they say, you know what? For the first time, I called up my friend. Or for the first time, I did some artwork, which I haven't done in like months. I don't think I've ever seen anything like this before. I mean, I do ECT and you see some remarkable changes, but not with this rapidity, not with this speed. Now, when it comes to the QIDS, the self-rating scale, it tends to be a bit slower. We follow the official QIDS, questionnaire, which is, can you tell me the average of your mood over the last 7 days? So I think that has something to do with it. The patients are averaging out their mood state. So it's a bit slower, but still within the first treatment, we're seeing a 22% reduction after just 1 treatment. By 3 weeks at the end of our treatments, the response, that is individuals with 50% drop in MADRS or the QIDS, very high, about 79% actually filled out those questionnaires or answered those questions that way. When it comes to actual remission, that is less than 10 on the MADRS, less than 6 on the QIDS-SR, around 50 to 70%. Again, very similar to the kind of numbers that we're looking at in the IV world. Now realize though that this is somewhat of a white glove approach, right? These are patients who come into the hospital. When it comes to psychedelic treatments, mindset, and the setting in which you give it always has a role to play. So that's one thing that I'm well aware of, that this is 3 weeks in hospital, nurses, doctors seeing you every day, and there's clearly always a psychological component to that experience. Now, just quickly, when it comes to the dissociative and psychedelic experiences of ketamine, my impression is that this is very idiosyncratic. We do what's called the CADS scale, CADSS. This is the clinician administered dissociative symptom scale. And basically what you see is that when we jump from the 0.25 to the 0.5 milligram per kilogram dose, that dissociative experience definitely increases. The kinds of experiences you see is quite interesting. A lot of patients will actually describe visual changes during the treatment with ketamine. If a person's wearing a patterned shirt, the pattern will start waving. The edges of a table will look blurry. Time sometimes slows down for individuals. So there's a psychological effect to it. So during the ketamine experience, patients tend to talk more disinhibited. The neat thing about that, of course, is that sometimes even without provocation, patients will say, I'm starting to remember something that I didn't think about for a long time. A patient came to me a few months ago and basically said, you know what, I remember when I started having depression, my dog died. I haven't thought about my dog in months. And these ideas will spontaneously come to them. Not in the same way of psilocybin or LSD, right? Not to massive mind-blowing expansion kind of way, but a more subtle way, which I think can be utilized during the treatment itself for psychological and supportive work. Now, as for the classic psychedelic experience, the entheogenic experience, right? Feeling God's presence, or the empathogenic experience. I feel the world and the universe, and I am one with you and everybody else out there. Or the entactogenic effect, that I'm moved internally, emotionally by some kind of force. We don't see as much of that with ketamine, but I got to say that, at least a couple of the patients had a fascinating experience. They said, I'm sitting here with you and I feel that we are one. I feel that, you know, the universe is one. So you do get some of those kinds of experiences with ketamine. One thing that we want to differentiate between ketamine treatment and some of a more aggressive psychiatric treatments like ECT, of course, is cognition. And this is, I think, one area, not only if ketamine works, not only is it relatively quick, but it's free from cognitive side effects, right? Whereas ECT, after 10, 12 treatments, patients will describe a general psychomotor slowing, memory deficits are pretty common. You don't see that with ketamine. Now, obviously, we're doing these computerized cognitive tests basically 3 weeks apart. So there's going to be some practice effect for the tests being done, you know, at a short duration. But basically, you're seeing what you would hope to see, that generally there is an increase in cognitive ability described, even if it is just a practice effect in that case. Now, in terms of safety, the main thing with ketamine, it's extremely safe. In fact, in the third world, they use ketamine even without monitoring sometimes because, you know, if monitoring is not available or pulse oximeter is not available, because ketamine does not cause respiratory depression. If anything, it actually, at least for us in psychiatry, because it creates that increased burst of energy, you would see blood pressure as well as heart rate increases. But the increases are not that severe and they're relatively easy to deal with. Nonetheless, we do monitor the vital signs every 15 minutes to make sure that we're not missing some poorly controlled hypertensive reaction. All right, now the next step, right? So this is just our inpatient side, which we've been doing since 2021, and that will continue. You know, for the most treatment refractory, severe depressed patients, I'm happy to take referrals and admit patients and work with them. But we want to be able to move this forward and see whether we can start treating outpatients as well. And that's what our ketamine intervention program is about. Hopefully in the next month, we will be able to take referrals for this program. And we're basically going to use this protocol from the inpatient world to the outpatient side. 6 treatments again, twice a week. And let's see what kind of results we get without, you know, that white glove inpatient treatment setting. Here at UBC, it'll be myself and Dr. Ray Lam heading that project, and Dr. Wynn in Squamish General Hospital will be running the cases through Coastal Health and on the and probably some cases through the Sunshine Coast. We are going to open it for residents of Vancouver Coastal Health, referred through community and also hospital psychiatrists. 6 treatments in total, 2 times per week, like we did before. Inclusion, exclusion will be very similar to what I showed a little bit earlier. And we can think about also maintenance treatment for patients, maybe up to 6 months. Obviously, given the limited number of slots, we want to make sure that's not too much of it gets taken up by maintenance treatments. All right, hopes and plans ahead. And I think, you know, this is where I'm going to close off pretty soon. First of all, we want to create the infrastructure for a treatment like this, because who knows, down the road, there could be others. Psilocybin, for example, has a lot of interest behind it, although there are some very clear technical difficulties of that compared to ketamine. We want to be able to compare subcutaneous versus intravenous, the typical IV ketamine infusion technique. We want to look at the duration of action, because unless we know how long this acts, is it 1 month, is it 2 months, is it 3 months, then we don't know how we're going to best schedule this for maintenance treatment. For example, can patients maintain their depression-free state if they came in once a month, for example? There are opportunities, of course, to look at the mechanism of action. You know, I've hinted at a little bit earlier, but, you know, for you guys out there, the researchers, we are certainly open for collaborations when it comes to this, all right? I can certainly imagine a lot of potential opportunities there. Ways to increase efficacy as a clinician is one that I've, you know, been wondering about, listening to these stories and sitting with these patients during these treatments. And I'm going to borrow a word from the theology world. The word is metanoic effect. Basically, what that means is during these experiences, however we define it, whether it is the change in functional connectivity of the brain, how do we engage in people so that they can actually change and have, whether it's a reawakening, whether it's inspiration, you know, those words that we use when we have peak experience in our lives where we just wake up and realize, you know what? I really like that person. You know what? I'm going to change my life. You know, it is those kinds of things that would be, you know, remarkably powerful in psychiatry if we could harness that potential. And then finally, exploration of ketamine treatments in the algorithm of refractory depression. You know, already there have been papers looking at ketamine versus ECT. That's how quickly people jump, right? You're not doing ketamine versus TMS. We're talking about ketamine versus our gold standard. Literally just 2 weeks ago, New England Journal of Medicine, there was this paper looking at 400 patients. It's an open label trial, so you got to take things with a grain of salt. But basically, they compared non-inferiority of ketamine twice a week, like what we're doing, versus ECT. And of course, they were using the IV standard treatment. And they basically showed that, that the ketamine response was quite good, 3 weeks and 6 weeks down after treatment, sort of like ECT. All right, in summary then, okay, psychedelic treatments are here. They are back. Clearly in the literature, they are growing in number. A lot of it is still obviously in the research world, but we want to be able to take it into the clinical world. 20 years now since that Berman paper in 2000 looking at ketamine, and it's really hard to access other than in private facilities or the very expensive intranasal Spravato S-ketamine treatment. Ketamine is novel in that it uses the glutamatergic system, right? The NMDA antagonist system, not SSRIs, not dopamine, not noradrenaline. There are good reasons to expect that we can have more than 50% treatment refractory depression patients at least respond, at least somewhat improve, despite being, 24 months with depression now. And at VCH, we're hoping to increase the opportunities for treatment of these patients through this ketamine intervention program, which hopefully you'll hear more about in the next month, certainly for the clinicians. All right, thanks to the team. Thanks for the leadership in the regional as well as the hospital program, for the operations people. And I gotta give a special shout out to our nurses on One West with being keen to try something new during the pandemic and now afterwards. All right, just gonna end off with a few papers. Top 3 are basically reviews of ketamine, the way you do it, the treatments, and how you set it up. This quick little paper, this was when we had 10 cases using our subcutaneous technique. Now we've got 24, and I've showed you the data on 24. But if you want to read more about it, there's links to the protocol and things like that. This is the ketamine versus ECT for non-psychotic treatment of major depressive disorder in New England Journal of Medicine that came out a couple of weeks back. And finally, this last one, if you're interested in the cutting edge of psilocybin, this is what they do, right? Often 1 or 2 doses of psilocybin, but psilocybin takes 6 to 8 hours, okay? A ketamine treatment is about 1 hour in duration. So that is a very clear methodological limitation of psilocybin, at least right now, at least until they can hopefully find out some new agent that can last a lot shorter in its effect. All right, so, and that is that. If there are any questions, I'll just leave it on this. If there are any questions, feel free to ask.
So Dr. Franco had to leave early, so we do want to take a couple of questions. Sure.
Doesn't work? Oh, but you can hear me. Okay, great. So thank you for this great talk. So I have actually 2 questions. So I would like to just know your opinion about topic that you discussed about dosing, because I was listening to a couple of weeks ago, there was a great podcast with Dr. Kacharis, and here he was discussing this, that in treatment using psychedelics in general, that we should use those that actually are feelable, like instead of using micro dosing. But you are showing that you have some effect with those that people, by micro dosing, I understand that those that people don't, that, like you said, that people said, oh, I don't feel anything, right? That will be the micro dosing. And you like showing that we've increased the dose, like 0.5 milligram, they start to report some effects. And do you have do you follow people just on this 0.25, or do you increase dosage for all your participants?
Yes. So excellent question about the dosing. Very important indeed. Now, I think there are some differences depending on what indication you're using it for, right? So, for example, when we think about psilocybin, in terms of the treatment for psilocybin, other than that last paper there, looking at depression for psilocybin, a lot of the studies will be around traumatic experiences, right? PTSD, maybe used in patients who are palliative and there's severe anxiety, dysphoria around death. So for those kinds of treatments, you can imagine that the psychological effect does need to be a bit higher, right? Because for psilocybin, for example, if you're treating somebody with psilocybin at a good dose, which is something like 25 milligrams, you literally have to hold their hand and watch for a really bad trip, right? You don't have to worry about that with ketamine for the most part, because the kinds of doses we use will generally not result in a bad trip. And even if that were to happen, the half-life is short, not like the 6 or 8 hours of psilocybin, and you can abort it with some moderate to high dose benzodiazepines. So there's that. So when it comes to dosing, yes, microdosing probably doesn't work, at least when it comes to the research literature. I know a lot of people swear that it works. 1 milligram of psilocybin, or even less than that, even like 0.5 or 0.25, some people say will work. I haven't seen good research on that, but usually with the protocols that have been published, we're looking at good doses where patients actually experience it. Okay, and I think it's slightly different from the ketamine because we're doing parenteral, and on top of it, we're aiming for a depression effect, not so much a psychological effect.
Great, thank you. Can I just ask one more question? Because you like showing those differences pre and post in cognitive measures. Do you have like a team studying, for example, like the electrophysiology, like EEG or other neuro imaging techniques pre and post?
That's why we want to open it up for collaboration.
Okay, we should talk.
This is, so far we have been running this on the edge of my table with my co-conspirators, as I mentioned, right? The anesthesiology people, pharmacy people, and basically using what we know out there in existing literature to replicate something that's feasible, that's doable. And that's kind of been, my level of looking at it right now. Absolutely. I mean, if there is a way to look at these patients before, after, use, neuroimaging techniques, EEGs, more detailed cognitive testing, we'd certainly be open for that kind of collaboration. And one more thing, by the way, ketamine, if you give it really quickly, okay, like what we're giving is like 0.5 milligrams per kilogram. So for a 70 kilogram man, we're only looking at about 30 milligrams or so. The drug users go up to 100 milligrams, 150 milligrams for the real extreme ones, okay? That's where they enter the K-hole. That is that is a sudden state of almost catatonia, near-death experience, that level of psychedelic experience. So just to give you an idea, what some people use, we're relatively small doses in comparison. Yes, at the back there.
I have two questions. Once they are improved by 2 weeks trial, then how long the improvement lasts by your experience? And my second question is, in treatment refractory patients, once they are improved like ketamine, is there any possibility that they become responsive to conventional antidepressants?
Okay, yeah, that's a great, interesting questions. So first of all, in terms of duration of action, hard to know. The research around repeated ketamine, like for example, a regular schedule twice a week, 6 treatments, seems to show that the majority of patients remain depression-free about a month down the road. 6 weeks down the road, or 2 months or 3 months down the road, we don't know. And that's why, part of our project here will include looking at the longer term. We'll try to, after our treatments as an outpatient, we'll try to follow them. I don't have the ability to do that right now on the inpatient side because most of these patients are not my patients. Now, the ones that I have followed, okay, and I've probably followed about 10 of the 30 or so, it looks good. Out of the 30 treatments that we've had over the last 2 years, I've had to bring 2 back because they felt that it was beneficial. And after about 3 months or 4 months, the effect kind of pooped out and they needed, like a maintenance treatment. But I've only brought them back like a year later or a year and a half later, not immediately afterwards. So I'm encouraged by that. But remember, this is a lot like ECT, right? You give 12 treatments of ECT. How long does it take before the person's mood go down? Unless you put them on good antidepressants and say lithium or something like that, usually within 6 months, the number of patients that become depressed again becomes substantial, right? 50% and above. So I would imagine it would be the same kind of thing. You improve the mood. For those who are very refractory, they will drop out after 3 months or 6 months or so. The second question, sorry, I kind of missed that one. What was that?
Is there a possibility that they become responsive to conventional antidepressants?
Responsive to antidepressants? Wow.
Yeah, if they are responsive to ketamine, is there any possibility that they become responsive to conventional antidepressants?
Right. I don't know. I don't know if there's any research looking at responsivity or retrials, for example, of a medication that they failed before after ketamine, and maybe it's more responsive or more able to maintain their mood state. But that's a great question. Yes. Hello. Yeah.
When you administer the ketamine, what does the session involve? Like, do you deliver a sort of like boosted therapy session, so it's like a normal psychological kind of thing, but just without the help of ketamine? Like, what does it involve?
Yeah, actually, no, it doesn't. And we're not. If you look at the literature, you'll see a lot of ketamine-assisted psychotherapy programs, right? The KAP is an often acronym that they use. No, for what we do, we actually bring the patients into on the inpatient ward into one of the rooms that we have. We can do 2 treatments at one time, 1 nurse, 2 patients. They're seated there with the monitoring equipment. And then the anesthesia nurse, who is trained for parenteral treatment of medications, will come with the ketamine in a needle and will give it subcutaneously, usually in the abdomen, okay, to allow it to, over the next hour or so, absorb into the system and create its effects. For some patients, when there's a one-to-one opportunity, I will actually go in and do some of that psychological work, mainly supportive, mainly trying to bring up and talk about some of the issues that might have brought them into hospital that might be related to the depression. Could be all kinds of things, right? And I mean, it's rife with possible discussions, whether it's relationship issues, family issues, whatever, grief. In those sessions, I will kind of select them for individuals that seem to be more responsive to want to talk about those situations. But the vast majority of the cases that you see here are not with therapy. They are perhaps with therapy afterwards. They're done there 1 hour, the monitoring's complete, they go back to the ward, and then the afternoon, within a couple of hours, I'll touch base with them and sit with them. I do tell my patients, though, that since there is this potential for you to have an interesting or unusual experience, why don't you, during that time, listen to some relaxing music? If you are one into meditation, why don't you do something like that? If there is something in your mind that you're really trying to figure out and tease out, why don't you think about that? And maybe in that experience, you will find some answers. And yeah, some patients come back and say, look, in my experience, I guess I found what I loved in terms of creativity. People who draw pictures and do art, that is a source of inspiration for them. And they sometimes will do it afterwards.
So you kind of leave it up to them to like explore themselves.
Yes.
So it's no risk that afterwards they go on to like try and self-medicate and end up using the drug themselves.
Yeah, absolutely. And that is why we try to have that exclusion criteria of significant substance use. Again, in the clinical world, we don't want to exclude people who could benefit. And given that we're a small study right now, small clinical project right now, we've put in that criteria of 6 months substance use free, right? And hopefully that will not result in any issues, but that's certainly something we'll keep monitoring down the road.
So 2 questions, maybe following up on that one. I understand the substance use exclusion criteria for a small trial and the risk that was mentioned. Is there any research on this as a potential treatment for substance use? Particularly thinking of this experience that you mentioned about, you know, sudden insight, sudden change in perspective that can be a part of it.
Yes.
Then I can.
Okay, sure. Absolutely, absolutely. You know, look, if you look at the reviews of ketamine usage in substance use, I believe there have already been 6 studies looking at the use of ketamine in alcohol use disorders. There are others in I think cocaine abuse as well. Now, to be honest, I haven't looked too much into that literature as to how exactly they do it. You know, do they do it like what we do at 2 times a week, 6 treatments, or do they have some kind of other modality? But yes, ketamine has been used in that literature, and you know what? That it's important. I'm glad that you bring that up, because the treatment of substance use has always been a thread in the psychedelic research from the 1950s and 60s. You know, I believe the founder of AA went through LSD treatment before he came up with the 12-step manual. You know, these therapies have that potential, you know, and we have to be cautious about going forward with that. But definitely there's a thread in that. And maybe in the days, I know Christian Schutz, researcher, many of you know him here, you know, he's got a protocol in the use of psilocybin, for example, for treatment of substance use disorders. How we can implement that, of course, you know, that remains in question. But yes, we're keeping an eye on that.
And the second one is there, where is the research again, or the evidence on bipolar depression? Are there any risks? Are there any benefits?
Yeah, studies on bipolar depression looks good. Actually, you know, again, this is one of those underserved areas where we don't have very many good medication treatments. So the studies on bipolar disorder with ketamine only show a very small percentage. I think it was less than 10% risk of creating a hypomanic or manic state. So it is potentially very good. And in fact, 2 of my cases were bipolar depression. Oh, way at the back.
During your study, have you come across any studies working on when it comes to depression, it is the broadest spectrum, and it can be seen in different diseases like mentioned briefly mentioned. Have you come across studies working on these things specifically as a treatment for depression in these types of diseases?
Yeah, you know, unfortunately, right now, I don't think I've ever come across a study with any substantial numbers other than case reports, right? You know, for example, I've got 2 patients with multiple sclerosis, very treatment resistant, and both of them did very well on the ketamine treatment. I think the main thing is, so long as there are no medical contraindications, you know, so I mentioned that 2 cases actually had strokes. You know, one of them actually was because of a hypertensive crisis from, I believe it was cocaine that the individual utilized. So we had to be very cautious about that case, because the person actually had a bleed, but it happened to be many years ago. Blood pressure is well controlled now, and even though there's an old hemorrhagic stroke, the person is doing well. So long as there are no major medical contraindications, I think definitely we can look into those populations. And I think that area is totally open for research.
Great, thanks. I think we're running a little bit behind schedule now, so thank you to Dr. Tan.
Pleasure. Let me just close off my computer.
So I'm going to pass it over to you, Dr. Murphy, to proceed with the awards.
Thank you, Claire. This has been really fun. Being involved with Research Day, and I guess I also want to thank all of you for voting. It's sort of a People's Choice Awards. We used to have to go and wrangle judges, and then now we just do it all electronically. So this is really quite smooth. So we're going to give awards for first the lightning talks. We're not breaking them down into clinical or basic or anything or translational. We're just going to have the first set that Claire was hosting in early, and for set A, we have Hallie Scherer and as winning, and this presentation was about movie watching as an alternative to resting state MRI. So this is Hallie. Come on down, get your award. Yeah.
Thank you very
Nice
much.
job.
Thank you.
And
Thank you so much.
okay, and for the second group, lightning talks, we have Rebecca Zivanovic, and this was on contraceptives and advice for mental health patients. So Rebecca.
Thank you. Thanks so much. Thank you.
Now, posters were broken up into a clinical and basic translational. And to start out in the clinical posters, we have Tria Ramburn. And this was an interesting poster about community gardens as a psychosocial intervention for refugees and migrants. Nice job, Tria. Okay. And lastly, in the basic translational category, we have Borna Atrian, and this was for the Minder app. This is an e-intervention for secondary school students or mental health. Well, thank you, and I'll turn it over to Joseph.
I think by virtue of being the last person to speak, I get to hand out some awards, which is cool too. All right, so we've got some undergraduate awards for medical students. First of all, there is J.C. Leung Memorial Prize, which is awarded to a fourth-year medical student who has demonstrated academic excellence in psychiatry. And for this year, it's been awarded to Jake Johnston. The Jake's around. We'll distribute these to the students who are not here. Secondly, we've got the Kristen Sieverts Memorial Prize in Medicine. awarded to a third or fourth year medical student who demonstrates development of the core competencies of an exceptional physician early in their medical training. And this year it goes to Miriam Manna. All right, thirdly, there's the Annette E. Hacking Memorial Award in Medicine and Psychiatry, awarded to a student with an aptitude for and interest in a career in mental health. Adjudication is based on leadership skills, commitment to community service, and demonstrated social conscience. This year it goes to Myrna Henry. Okay, the Andrew Neil McTaggart Memorial Prize in Psychiatry, awarded to a medical student who has demonstrated interest and academic achievement in the study of child and adolescent psychiatry, and is made on the recommendation of the UBC Department of the Division of Child and Adolescent Psychiatry to Jenny Sang. Come on down, Jenny. All right, let's take a picture. This is yours. Okay. And to round out the undergrad awards for medical student, it's the Dennis Harris Memorial Prize in Psychiatry, awarded to a third-year medical student who has demonstrated academic excellence in psychiatry, to Lance Shaver. Lance, Tim, I think you have the graduate awards. I think you're back up here with the graduate award, according to my script here. Just leave that.
Happy to do these. Okay, for postgraduate awards, we have the George A. Davidson Scholarship, and this is awarded to a resident who has demonstrated exemplary research and scholarly activity during residency training. And this is given to two people, to Allison Hudson and Maria Alexandra Angel. And then we have the CanMeds Award. This is a peer-nominated resident who best exemplifies the CanMeds competencies of medical expert, communicator, collaborator, manager, health advocate, scholar, and professional. So we have a series of awards here. First place goes to Alexander Levitt and Victor Lee. Okay. Second place, we have Jordana Wasserman and Quentin Clark. Yeah, why don't you come down for a picture? I'll just list out the rest of the awards. There aren't plaques, I just mentioned. We also have Oshin in third place, Oshin Maharashi Wari, sorry. And in fourth place, we have Adib Malas and Max Liu. Fifth place, we have Shika Walia and Faizan Batia. We also have an award for Resident Undergraduate Teaching Award, and this is honoring residents who have had significant positive impact on the learning of students in the MD undergraduate program. And in the Vancouver track, we have Tristan Willis. will receive a plaque. In the Fraser track, we have Faizan Batia, also receiving a plaque. Congratulations. Okay, for Vancouver Island, we have Connor Zier Wankland. Oh, so fast. Congratulations. Thank you. And lastly, in Prince George, we have Taylor Callender. Then we have an award for significant contribution to research, and this is awarded in recognition of a resident who has contributed significantly to the research goals of the program by participating in a research project, publishing and/or presenting. And we have a plaque for Laura Labonte. Congratulations, Laura. Okay, then we have another prize, the Emily Ellingson Prize for significant contribution to the UBC Psychiatry Residency Program. And this is a peer-nominated award recognizing the resident made outstanding contributions to the program that have resulted in improvements to training, professional development, and camaraderie amongst the residents. And Alexander Levitt. Okay, then we have a significant leadership award, and this is for residents that has demonstrated consistent and outstanding leadership within the residency program and/or community activities. And we have Isaac Roden receiving the award. Congratulations. Thank you. Thank you. Then we have the Dr. William Piper Memorial Award in Psychotherapy. And let's see, so this is awarded to a resident who has shown strong interest in psychotherapy, has exceeded standard expectations in acquiring psychotherapy skills, and demonstrated outstanding development as a psychotherapist. So we have Connor Hawkins and Alexander Levitt. Congratulations. We also have a 2023 Nasia Laura May Elvin Memorial Prize in Schizophrenia Research. And this is awarded to a PhD student or a postdoc trainee that has published an outstanding paper relevant to schizophrenia. And this one is awarded to Andrea Jones. All right, then we have the Island Medical Program awarded to, this is the Emily Ellingson Award, and this goes to Darcy Good. Okay. And we also have Providence Healthcare Department of Psychiatry Awards. We have the Dr. Brian Morris Memorial Award. This is a resident that has demonstrated exemplary compassion and empathy towards to patients and clinical excellence. This goes to Stephanie Montgomery.
Thank you.
Oh, here she is. Hi, Stephanie. And another Providence Healthcare Award, the Dr. Maria Corral Award. This is awarded to a resident that has shown excellence in teaching medical students during psychiatry rotations at St. Paul's Hospital. And this goes to Gail Lee Thompson. And another Providence Healthcare Award is the Dr. Robert Kitchen Memorial Award. This is given to a resident that has demonstrated excellence in consultation liaison psychiatry training at St. Paul's. And this goes to Connor McLean. We're down to our last few awards, and I realize that many of these are affiliated hospitals or island programs and stuff, so it's understandable that not everyone is here. So we also have the Dr. Megan Roberts Honorary Scholarship, and this is a special mention. There's no plaque, but this honors the legacy of our colleague, Dr. Megan Roberts, for her extraordinary impact on psychiatry residents, physician colleagues, and patients. And it's been established to recognize the Royal Columbian Hospital residents who demonstrate excellence in leadership, collaboration, mentorship, all the qualities embodied by Dr. Roberts throughout her life and career as a brilliant clinician. So this goes to two people from the Royal Columbian, and this is to Daniela Crocker, and then Daniela, and then also to Jessica Chin. Congratulations.
Thank you.
I think we're out of awards. I see it as a great honor to present these awards. I know Lakshmi usually presents them, and it's great to become familiar with the residents and all these different sites and great programs. And the talks today, wow, I mean, hearing about the ketamine clinic and its effectiveness and all these new tools, using movie watching as a stimulus instead of resting state, many different community gardens as a therapy, a lot of creative things, apps for high school students to assist them in mental health. I think the department is really rising above, and this is really cool work. And I want to thank all the presenters and people who came and voted on Slido. And I don't know if there are any other closing remarks, but let's just give a round of applause to
let's go talk to this guy.